As we age, our blood becomes less like a sleek high-speed rail delivering precision-timed immunity and more like a cluttered baggage train stuck in delay, carrying tired immune cells, inflammatory debris, and molecular signals of past biological battles. This slow, subtle breakdown of blood composition profoundly shapes how our immune system functions in later life, giving rise to a phenomenon known as immunosenescence. The culprits are many: exhausted T cells, sluggish stem cells, rogue cytokines, misplaced antibodies, and a creeping tide of chronic inflammation that turns the bloodstream from a sanctuary into a slow-boil stress bath. Together, they form the basis of the blood-immune aging axis, a deeply entangled feedback loop that helps explain why older adults are more vulnerable to infections, cancer, autoimmunity, and yes, bad outcomes from even ordinary colds. Let's start with the T-cell repertoire, those frontline agents of adaptive immunity trained to identify and destroy invaders with surgical precision. In youth, the thymus pumps out fresh T-cells like a startup flush with energy. But the thymus shrinks dramatically after puberty, a phenomenon called thymic involution, and with it, our ability to generate new naive T-cells plummets. What's left are memory T-cells, which have seen their fair share of pathogens and now operate more like grizzled veterans with limited adaptability. In the blood of older individuals, we find an accumulation of senescent T-cells, particularly CD8+ cells that no longer proliferate or function properly, but still secrete inflammatory molecules like IFN- and TNF-A, adding to the background noise of inflammaging. Think of them as retirees who refuse to go home and instead linger in the office, slowing everyone else down while muttering about old battles. But it's not just the T-cells themselves that change, it's the stem cell environment that feeds them. Hematopoietic stem cells, HSCs, in the bone marrow are responsible for generating the immune cell supply chain. As we age, these HSCs become less efficient, more skewed toward producing myeloid cells over lymphoid ones, and more likely to generate cells with inflammatory tendencies. This shift is reflected in blood: a higher neutrophil to lymphocyte ratio, reduced B-cell diversity, and increased production of monocytes that contribute to a low-grade inflammatory state. In other words, the blood gets older, and the immune system starts to look less like a coordinated army and more like an overstaffed bureaucracy filled with paperwork and miscommunication. And speaking of paperwork, let's talk about cytokines, the molecular messages that coordinate immune responses. In aging blood, there's a well-documented rise in pro-inflammatory cytokines like IL-6, IL-1S, CRP, and TNF-A. These are not the sharp, specific responses that resolve infections. They're the background hum of discontent, fueling chronic diseases like atherosclerosis, type 2 diabetes, and neurodegeneration. This cytokine fog doesn't just impair the immune system, it exhausts it. B cells become less efficient at producing high-affinity antibodies. Natural killer, NK, cells lose their killing capacity. Dendritic cells struggle with antigen presentation. It's as if the immune system loses its memory and its aim at the same time. What's especially troubling is that this dysregulated immune state feeds back into blood composition itself. Damaged cells release DAMPs, damage-associated molecular patterns, which enter the bloodstream and perpetuate inflammation. Senescent cells, which increase in tissues throughout the body with age, release SESB factors into circulation, further degrading immune control. Even the clonal expansion of dysfunctional immune cells, a phenomenon seen with aging, leads to a kind of monoculture in the blood, where immune diversity shrinks and the system becomes more vulnerable to novel infections and vaccine failure. The consequences are everywhere. Older adults respond less effectively to vaccines, not because the antigens are wrong, but because their immune systems can't generate robust memory responses. They're more prone to reactivations of latent viruses like CMV and VZV. Cancer surveillance declines. Wound healing slows. Even psychological stress can have a bigger immune impact because the hormonal stress response, e.g., cortisol, intersects with immune function via bloodborne signals. The blood has become not only the battlefield but also the bottleneck. Yet it's not all doom and decline. Emerging research suggests we might be able to modulate the blood-immune axis to rejuvenate immunity. Interventions like caloric restriction, exercise, and fasting mimicking diets have been shown to reduce pro-inflammatory cytokines and improve immune profiles. Experimental approaches like senolytics, which clear senescent cells, or parabiosis-inspired plasma exchange, removing aged plasma and replacing it with youthful equivalents, are showing promise in early studies. There's even interest in rejuvenating HSCs or reactivating thymic function, potentially restoring a more youthful immune tone through internal rewiring. So while aging blood undoubtedly contributes to immune decline, it's also an accessible, measurable, and potentially malleable target. Blood tests can now reveal more than cholesterol and glucose. They can paint a picture of biological age, immune readiness, and inflammatory load. As we learn to read this picture more accurately, we may also learn how to revise it through lifestyle, medicine, and maybe even next-generation therapies that rewrite the aging script molecule by molecule. In the end, your blood doesn't just reflect the age of your immune system, it shapes it. It's not just the road the immune cells travel, it's the landscape, the weather, the traffic report, and sometimes the danger sign. Understanding that could be the key to a healthier, longer immune lifespan and maybe, just maybe, to turning your inner immune world from an aging battlefield back into a well-run, youthfully sharp defense force.